Hey everyone, and welcome. Today we're diving into something really cool. The story of how scientists figured out how to describe the tiny engines of life, our enzymes, using the language of math. We're going to see how just a few key equations can unlock the secrets of these amazing biological catalysts, how fast they work, what makes them tick, and even how we can stop them in their tracks. So here's our game plan. We'll kick things off with a pretty basic question. Why should we even bother trying to measure how fast an enzyme works? Then we'll get into the big one, the Michaelis-Menten equation. We'll see how to actually visualize it and then use that knowledge to understand how enzymes can be blocked or inhibited. And finally, we'll wrap it all up by seeing what this whole mathematical toolkit really tells us. All right, let's set the stage. We all know enzymes are the absolute workhorses of our cells, right? But just saying they're fast or efficient isn't really enough for science. To really get a handle on them, we have to go beyond just describing them and start actually measuring them. We have to turn biology into numbers. And at its heart, that's what enzyme kinetics is all about, answering this one huge question. It's about putting a number, a hard value, on the speed of life itself. We're quantifying the rate of all those chemical reactions that are powering every single thing your cells are doing right now. But here's the thing, an enzyme's performance isn't set in stone. It's a lot like a car engine. Its efficiency totally changes depending on the conditions. If you raise the temperature, the reaction speeds up, but only up to a certain point. Go too high and the enzyme literally falls apart and stops working. So to understand and predict this kind of behavior, we really need a mathematical model. And that brings us to the first major attempt to build that model. This is the cornerstone of biochemistry, developed more than a century ago by Leonore Michaelis and Maud Menten. Okay, so let's break down how their model works. It assumes this really simple three-step process. First, you have the enzyme, E, and the thing it works on, the substrate, S, and they come together. Next, they form what's called an enzyme-substrate complex, or ES. Now, this step is reversible. The substrate can bind and then just pop right back off without anything happening. But if it sticks around, the enzyme does its job, converting the substrate into the product, P. Then it releases the product, and the enzyme is totally free to start the whole cycle over again. And this is the hero of our story, the Michaelis-Menten equation. It's just a brilliant little formula that connects the initial speed of the reaction, which we call V, to the concentration of the substrate. That's what the brackets around the S mean. And it does this using two very special constants that are unique to every single enzyme. So what are those two constants? They're Vmax and Km. Vmax is pretty straightforward. It's the enzyme's top speed, pedal to the metal, the absolute fastest it can possibly work when it's drowning in substrate. But Km, the Michaelis constant, that's where the real magic is. It tells you the exact concentration of substrate you need to get the enzyme working at half of its top speed. It's basically a measure of how badly that enzyme wants to bind to its substrate. And here's the key takeaway about Km. It's really all about the enzyme's affinity for its substrate. You got to think of it as an inverse relationship. A really low Km means the enzyme is a strong grabber. It only needs a tiny bit of substrate to get to half speed because it binds so darn tightly. On the other hand, a high Km means it's a weak grabber, and you need to flood it with a lot more substrate just to get it going. Now, an equation is great and all, but you know what they say? A picture is worth 1,000 words. So how do we actually visualize what this equation is telling us? And maybe more importantly, how can we use that picture to figure out Vmax and Km from real lab data? Well, when you actually plot the reaction speed against the substrate concentration, you get this beautiful sweeping curve called a hyperbola. See how at low substrate concentrations the rate just shoots up? But as you keep adding more substrate, the enzymes start to get totally saturated, and the rate begins to level off, getting closer and closer to that top speed, Vmax. But you can see the practical problem here, right? How in the world do you figure out the exact value of Vmax from a curve that never actually reaches it? You're always just kind of guessing where that flat part is. A curve is just tricky. There has to be a better way to do this. And of course there is. In this amazing moment of mathematical genius, two scientists, Hans Linneweaver and Dean Burke, realized that if you just flip the whole Michaelis-Menten equation upside down by taking the reciprocal of both sides, you can rearrange it into the equation for a straight line. You know, the classic y equals mx plus b you learned in algebra? It's an elegant little trick that turns that tricky curve into something incredibly useful. And this is the result, the Lineweaver-Burke plot. As you can see, it's a perfectly straight line. So now, instead of trying to eyeball a curve, we can just draw a nice, clean line through our data points. And check this out. The y-intercept is just 1 over Vmax, and the x-intercept is -1 over Km. It's such a simple, powerful way to pull out an enzyme's most important stats. No more guesswork. Okay, so now that we have this fantastic tool, we can start to ask more complicated questions. Like, what happens when something gets in the way of an enzyme and messes things up? Well, these interfering molecules are called inhibitors, and understanding them is absolutely fundamental to things like designing drugs and figuring out diseases. First up, we've got competitive inhibition. In this case, the inhibitor molecule looks a whole lot like the enzyme's normal substrate. It's like a game of musical chairs. Both the substrate and the inhibitor are fighting for the exact same seat, the enzyme's active site. And the rule is simple: only one of them can sit there at a time.
So how does this competition actually show up in our data? Well, think about it. You can eventually beat the inhibitor just by flooding the system with so much substrate that the inhibitor barely gets a chance to bind. That means the enzyme's top speed, its Vmax, is ultimately unchanged. But because of all that competition, you need a lot more substrate to reach half speed, so the Km increases. And on a Lineweaver-Burke plot, this creates a classic pattern. The lines for the inhibited and the uninhibited reactions all cross right on the y-axis.
Next, let's talk about non-competitive inhibition. This kind of inhibitor doesn't even care about the active site. It binds to a totally different spot on the enzyme. This means the substrate can still bind just fine, but the inhibitor acts like a wrench in the gears. It messes with the enzyme's shape and makes it way less effective at actually doing its job. The kinetic signature here is completely different. Because some of your enzyme is essentially poisoned by the inhibitor, the overall top speed, Vmax, decreases. But since the inhibitor isn't blocking the active site, the enzyme's affinity for the substrate, and therefore its Km, stays exactly the same. This creates another really clear pattern on the plot. Now, the lines all intersect on the x-axis instead. And finally, we have this really unique case called uncompetitive inhibition. This inhibitor is, well, it's sneaky. It doesn't bind to the free enzyme at all. It just waits. It waits until the substrate has already bound, forming that ES complex, and then it swoops in and binds, locking the substrate in place and preventing the reaction from ever finishing. This really weird mechanism produces a very distinct result on our Lineweaver-Burke plot. It turns out that both Vmax and Km are reduced. And what this does is it causes the line for the inhibited reaction to shift up and to the left, creating a set of perfectly parallel lines. You can't miss it. It's an unmistakable visual fingerprint. So let's take a breath. After all of these graphs and equations, what's the big takeaway? What have we really gained here? Well, what we've gained is an incredibly powerful diagnostic toolkit. This table right here, this sums everything up. It's like a cheat sheet. By running an experiment, plotting the data, and just seeing what happens to Vmax and Km, we can diagnose exactly how a new drug or a poison is working. If Vmax stays the same but Km goes up, it's competitive. If Vmax goes down but Km stays the same, non-competitive. And if they both go down, it has to be uncompetitive. The math literally reveals the mechanism. And that brings us to one last, really provocative thought. The entire field of modern drug design is built on this very foundation. If we can use kinetics to understand precisely how an enzyme is being shut down, can we then turn around and use that knowledge to design from scratch a molecule that will inhibit it in exactly the way we want? The math doesn't just describe the problem, it actually points us directly toward the solution.